that suffer with chronic alcoholism actually affect almost all of their organs in the GI tract. So you'll see what some of the symptoms are with esophagitis, gallbladder disorders, pancreatic disorders, what can happen to the stomach, ulcers. So it kind of is a litany of different episodes that an individual can have with chronic alcoholism. So, all right, so along those veins, we are actually recording now. We did just go through the anatomy of the stomach. What I'm going to do is actually talk about receptive relaxation is a reflex that we have. It's a response that we have to eating a meal. So I just talked about the fundus, right? And basically what happens is with receptive relaxation, it describes a condition where relaxation of the fundus region precedes the arrival of a bolus into the stomach. So again, this is called receptive relaxation. As soon as you get basically some receptors that are activated, that's what this particular symbol is. These are mechanoreceptors right here in the fundus region that are detecting a stretch. So as soon as you actually start to eat a meal, these mechanoreceptors, you can see here, detect stretch. They send a signal via the vagal afferents to the central nervous system and back through the vagal efferents, which then causes the fundus to relax, to allow and accommodate more food. It's basically an anticipatory relaxation to accommodate more food that's coming down the esophagus. Okay, so vagal, whenever you actually see that term vagal, I want you to automatically think parasympathetic neurons. This is part of the parasympathetic nervous system. It's actually cranial nerve 10, and they're called vagal nerves. Okay, so automatically remember our parasympathetic nervous system is all about resting and digesting. Okay, so these are parasympathetic neurons. The relaxation is thought to involve an increase in potassium permeability, which results in a membrane hyperpolarization and a decrease in muscle tone within that area. And some of the neurotransmitters involved are thought to be vasoactive intestinal peptide, VIP, or ATP. So this is known as, again, one more time, receptive relaxation. So then once you get food in your stomach, it activates a number of different responses, mechanisms that cause basically the stomach to become a very violent place. It starts to secrete acid to chemically digest food. It also has these pacemaker cells, very much like the pacemaker cells in your heart, which gives a rhythmic type of peristalsis, circular smooth muscle, longitudinal muscle, oblique muscles actually twist the stomach, all helping with basically mechanical digestion. And then you see a type of phenomenon called retropulsion. And as you get this mixing within the stomach, the pyloric junction actually contracts, which may seem counterintuitive, but remember the GI tract is all about compartments. And so you want to be able to chemically digest that food in the stomach before you deliver it into the small intestines. So retropulsion occurs during the mixing phase, and it's basically a contraction of the pyloric junction, which propels food back up into the stomach, helping with the mixing process. So we'll talk more about the cephalic, gastric, and intestinal phases. I'll break it down into these different categories in just a little bit. This is more of an intro of the stomach. All right, so we've also talked about acid-base regulation a little bit. I'm going to give you a bit more information now. I want to talk briefly about vomiting. Like I said, the GI tract is not a glamorous place. Vomiting, the fancy scientific name for vomiting is emesis, okay? So if you actually took something that would help with nausea, a lot of times it's called an antiemetic, if you haven't ever seen that word. It's to help not vomit, okay? So what happens with you when you vomit, especially after you eat a meal? Imagine you eat a meal, you deliver all of this acid into your stomach, and then you vomit. So what occurs is basically something called a metabolic alkalosis. Metabolic alkalosis, which means your blood pH actually rises quite a bit because you've just vomited all this acid out of your body. Okay? So this is what happens with vomiting. You actually have a vomiting center in your brain. You first retch. Now remember, this is all about kind of biophysics. When you retch, you actually breathe in against a closed epiglottis. Everybody knows what the epiglottis is now, right? If you breathe in or try to breathe in with a closed epiglottis, it's something like this, right? So what happens when you do that is you actually create a negative pressure in your upper esophagus. Think about that. You're creating a negative pressure in the upper esophagus. And then a lot of times your stomach muscles, they contract. They contract your stomach actually, and they create a very high pressure in your stomach. And remember, things are always going to move from high pressure to low pressure. So as soon as that lower esophageal sphincter opens up, it literally propels food out of your mouth, from your stomach out of your mouth, okay? So remember, it's just about creating those pressure gradients. Now, this is actually very different than regurgitation. So a lot of people use these words synonymously, regurgitation and vomiting, but they are actually not the same. So when you talk about baby birds, right, baby birds eat from their mother's regurgitation. Regurgitation is actually reverse peristalsis. So you can imagine that there's a different mechanism, right? Peristalsis pushes the food back up through the esophagus and then into the beak. So vomiting and regurgitation are actually not the same, just so you know that, okay? All right, so fun stuff, vomiting. In that video, I'll explain more about metabolic alkalosis too. So don't worry, you will get that information. Okay, so let's take a look, let's zoom in. Actually, if you can take a look at the upper part of this diagram here, this square right here is actually being blown up. So we're zooming into the wall of the stomach. And you'll notice that the wall of the stomach is made up of different layers. The mucosa, submucosa, and smooth muscle layer. So this right here where you see the gastric pits, this is actually facing the lumen or inside of the stomach. And then as you move down these layers, you're moving towards the blood supply. All right? So the mucosa layer closest to the lumen of the stomach actually has these gastric pits. And within the gastric pits are these different cell types. Mucus neck cells, and as you can imagine, they secrete mucins. As I mentioned before, parietal cells, it's really important to know the name of these cells. Parietal cells actually secrete acid, hydrochloric acid. And then the chief cells secrete the protease pepsin. Actually, that's not correct. I'm going to be more specific here. Chief cells secrete pepsinogen, which is the precursor to pepsin. Okay, so chief cells secrete pepsinogen. And then these enteroendocrine-like cells secrete hormones into the blood. So these enteroendocrine-like cells can be G cells, which secrete gastrin. So now I'm going to put some things together. All right, so chief cells secrete pepsinogen, parietal cells secrete hydrochloric acid. That's responsible for the very acidic pH in the gastric lumen after you eat a meal. And here's another slide that really gives you more information about the crosstalk between chief cells and the molecules that are secreted by chief cells and parietal cells. So how is pepsin made in the stomach? Chief cells secrete the pepsinogen. Parietal cells secrete the acid. And it's actually the acidic environment that converts pepsinogen to pepsin. Pepsin is the active form of the protease. And then once pepsin is formed, it actually converts more pepsinogen to pepsin. That's kind of a positive feedback, right? To amplify the response. Pepsin is the active form of the protease, and it actually does break down proteins to smaller peptides. Now, one other thing that is really important about parietal cells is that they also secrete something called intrinsic factor. Okay, so I think we have a group here that is doing, talking about a disease called Pernicious Anemia. Pernicious Anemia. Get a pen out here. Switch over to the document camera. Pernicious anemia is actually a disorder if you're not able to secrete enough intrinsic factor, then it can cause something called, can I borrow your pen? Does anyone have an extra pen over here? Sorry. All right. Thank you. Yeah, no problem. All right. So the disease is called intrinsic factor. And it is actually a lack of, the disease is called pernicious anemia, and it's caused by a lack of intrinsic factor that usually is secreted by the parietal cells. Pernicious anemia. So what does intrinsic factor do? You might not know this, but is one of the most essential molecules for life. It actually helps you absorb B12, which is very important in red blood cell formation. So if you don't have this, you can get anemia. All right, so intrinsic factor. Just remember that's coming from the parietal cells. All right, so that's kind of listed here. You could easily, when you're studying, kind of miss it, but don't forget that parietal cells also secrete intrinsic factor. So let's zoom in on the parietal cells. We're kind of going from the gross anatomy, zooming into the wall of the stomach, and then zooming even further into the parietal cells themselves. Okay, so this square, let me give you some orientation here. This square is a parietal cell. So all of this beige area in here is a parietal cell. And it looks complicated, but you've seen this all before. Remember the reaction carbon dioxide and water is converted by the enzyme carbonic anhydrase to carbonic acid, which then is converted to bicarbonate and protons. Now, if you remember the kidney, those are parietal cells in the kidney. For every carbon dioxide molecule, one acid is secreted and one bicarbonate ion is reabsorbed, just like what we saw before. They're trafficked to different membranes. The protons are actually trafficked across the apical membrane by the the proton-potassium pump. This is actually a pump that uses ATP to move molecules against their concentration gradient. So this is actually one of those, don't get it confused with the sodium-potassium ATPase, but this is a primary active transporter. A lot of people call it the pump in the stomach, right? It is literally pumping protons into the lumen of the stomach, the interior of the stomach, and chloride follows. So I wasn't kidding when I say you literally secrete hydrochloric acid into your stomach after you eat a meal. Okay? The potassium channel here is just to make sure that this pump isn't rate limited, so it doesn't run out of enough potassium and it keeps pumping protons out. All right, so there's actually some pretty good drugs out there if you have heartburn, right? Heartburn. If you actually go to Walmart now and you actually see the purple pill, or I think that's called Nexium. The brand name, I'm sorry, not the commercial name, but the generic name is called Omeprazole. So take a look at that next time you're at Costco or something, and Costco will, they're not selling or they do, but they don't always sell the commercial name. You might actually see a box that says Omeprazole, okay? Omeprazole blocks this pump, okay? Omeprazole is a drug that actually inhibits that pump, the potassium proton exchanger. So acid production by the parietal cells in the stomach depends on the generation of carbonic acid, and it subsequently moves hydrogen ions into the gastric lumen, resulting from primary active transport. Now, at the same time, as you're pumping protons into the interior of the stomach, you're pumping bicarbonate into the blood. This is another reason why you become so alkalotic, which means your blood has a high pH after you eat a meal, because you've just transported a ton of bicarbonate into the blood, which is your major buffer. It's going to wick up any acid really that's in that blood. We're going to talk about how that travels to the pancreas and is delivered into the small intestines to neutralize any acid coming from the stomach in just a little bit. So the bicarbonate is actually delivered into the blood by the chloride bicarbonate exchanger. All right, pretty good? Everybody clear on that? Anybody have any questions? All right, so we're going to move on to regulation of the parietal cells. I want you, like I talked about this, the title of this lecture is actually hormonal control of digestion. So you'll notice at the top here, okay, before I get to that, this is supposed to represent a parietal cell of the stomach, right? Parietal cell. You can see here are our proton potassium pumps. They get inserted into the apical membrane and they secrete acid into the lumen of the stomach. You actually have one inhibitory and 3 stimulatory signals that ramp up acid secretion in the parietal cells of the stomach. Okay? Somatostatin, as you'll notice when we get even into the endocrinology section, just remember somatostatin actually inhibits everything. It really does. It just inhibits everything. So somatostatin in this case, is also inhibiting acid secretion from the parietal cells, whereas gastrin, that's a hormone. Histamine is a paracrine signaling molecule. Basically, this is coming from nearby cells, not from the blood. And acetylcholine, which is a neurotransmitter. All three of these ramp up acid secretion. So, how does that work? This is looks like a complicated slide. I'm going to go through it. I'm glad you all are here today because there's not much explanation other than what I give you right now. So remember, I am recording this. So if you need a little bit of help, you can go back and listen to this section. So basically what we have here is at the top, this is a parietal cell. And you can see the acid secretion. We're going to pretend that this blue part up here, this purple part, is actually the stomach lumen. So this is the interior of the stomach. So you can actually see protons are being pumped into the stomach lumen. We talked about our three stimulatory molecules. Gastrin is a true hormone coming from the blood supply. So this is supposed to represent a capillary. Histamine is coming from a nearby cell called enterochromaffin-like cell. I'll write that down. Enterochromaffin-like cell. Enterochromaffin-like cell. So that's why it's called an ECL cell. ECL cell. Okay, so if we go back, you can see here's the ECL cell. It's actually secreting histamine. And then our neurotransmitters, these are supposed to represent parasympathetic neurons. Remember our resting and digesting neurons. They secrete acetylcholine. Okay, so what the other term that I want you to understand is a term called potentiation. Potentiation. This actually really ramps up acid secretion. Potentiation. And this is how it works. Okay, so you can actually see once you start to maybe smell food, smell or see food, right? You already are going to get some parasympathetic neuron activation. And acetylcholine is going to be released, and you're already going to get, now, I hope you remember this one. This is a GS, GQ, or GI. If you increase calcium, GQ, right? GQ. So this is a typical G protein coupled reaction. It actually activates GQ pathways. You get an increase in calcium. That helps to insert those proton potassium pumps, and you get more acid secretion. And that's just called the cephalic phase. That's just a little bit of smelling or seeing food. You're already getting some acid secretion. Then you start to eat a meal, right? A little bit of food enters into the stomach. You get receptive relaxation. The fundus starts to relax. And then you start to trigger the body of the stomach. G cells actually start to secrete gastrin. Gastrin gets released into the blood supply and travels to the parietal cells. It is also GQ coupled. So you get more acid secretion. Now, here's where it really ramps up. You can see that gastrin and acetylcholine also work on and activate these ECL cells, enterochromaffin-like cells. Once there's acetylcholine activation, parasympathetic nervous system activation and acetylcholine release, and gastrin on board, circulating through your blood supply, that triggers the ECL cells to release histamine. Histamine is GS coupled. Now this is the trifecta. All three of them are on board and you get the maximum amount of hydrochloric acid secretion. So what potentiation means is if you just trigger the parasympathetic neurons, you're going to get a little bit of acid secretion. If you just have gastrin on board, you're going to get a little bit of acid secretion, or just histamine. But when all three are on board, again, this is the trifecta, the amount of acid secretion is greater than the sum of the individual parts. Okay? So again, I'll say that one more time. What potentiation is basically the amount of acid secretion with potentiation when all three are on board is greater than the sum of each of these individual molecules and what they acid that they produce. Yeah, question. I would say an additive effect almost. And then on top of that, you actually are getting even a little bit more. Yep. Okay, so that's known as potentiation. All right, so here's a flow chart. It's kind of telling you the different types of secretions. We've talked about saliva and salivary, the amylase. We've talked about acid secretion, mucus, bile. We haven't talked about bile yet. Bicarbonate and digestive enzymes. When we get to the small intestines, we'll talk more about that. But here's a nice kind of flow chart talking to you about, giving you some information about this phase where you just are looking and smelling food. You already get some acetylcholine activation, parasympathetic nervous system activation, and acetylcholine release. Parietal cells are now secreting some acid. Then with ingestion, you get those mechanoreceptors that actually trigger those G cells to secrete gastrin, which trigger ECF cells. These are ECL cells, enterochromaffin-like cells, and chief cells. More acid secretion. Chief cells now secrete pepsinogen, and so on. So this is telling you, it's kind of giving you more information about the global response. Here's where I break it down. You have the cephalic phase and the gastric phase. So the cephalic phase, if you want to write this on the margins, the cephalic phase is really just about seeing, smelling food. And when that happens, you can see those vagal efferents are already being activated and releasing acetylcholine, which are triggering those parietal cells. You're also getting a little bit of gastrin secretion through these G cells. You can see that some neurons actually release gastrin-releasing peptide. That's not very original. Gastrin-releasing peptide. It triggers those G cells to release gastrin, which then also plays a role in ramping up the acid secretion. That all happens in the cephalic phase. The gastric phase, now this is actually when you've eaten something. Now you can see these really weird looking M. These are supposed to represent mechanoreceptors. You're going to get fundus, the receptive relaxation, more acetylcholine release, gastrin starts to ramp up the system. You have these long reflexes. Long reflexes actually go all the way to the central nervous system by the vagal afferents and back through the vagal efferents. That's a long reflex system. But you also have these little short reflex systems, reflex neurons that are being activated too, ramping up this system. Okay, more gastrin, more acetylcholine. Now the ECL cells come on board. Histamine is released, and you get the maximum acid secretion. This is when the motility is really ramped up as well. Chemical, mechanical digestion. So if you need actually a table associated with this, you can actually see this is now breaking it down into the cephalic phase, the gastric phase, And in a minute here, we're going to jump into the intestinal phase. All right, more flow charts, but it's really saying the same thing. I'm going to skip over this. You guys can take a look at this when you get home. Remember, I also said that there's a lot of mechanical digestion going on here. We've got the circular smooth muscle, longitudinal and obliques. Peristalsis, the system is really ramped up. And then we get into the intestinal phase. Okay, this is the intestinal phase. This seems a little counterintuitive, so I really want you to kind of listen to what I'm saying here. There's something known as gastric emptying. Gastric emptying. What that means is basically like it sounds. Gastric emptying is when you deliver food partially digested from the stomach into the small intestines. Now, what I want you to realize is it's not about a big dump, right? It just doesn't stop and you just dump everything into the small intestines. What happens is as soon as you actually get a little bit of food into the small intestines, it decreases gastric emptying. See, that seems a little counterintuitive. Why would you slow everything down? Why would the pyloric junction now increase contractions, right? So I want you to know these sensors here that are sensing fats coming into the small intestine, acid, and solutes are now going to feed back to the stomach and slow gastric emptying, decrease gastric emptying. Because now it's just about piecemealing a little bit of food at a time into the small intestines so you can maximize digestion and absorption. So we're just going to, now it ramps everything down, acid secretion goes down, peristaltic contractions go down. And the idea here, and I can guarantee this will be a test question, the intestinal phase decreases gastric emptying. Okay, so here's where a lot of hormonal control occurs. A lot of hormonal control. Again, you have these sensors within this area. These sensors are detecting fats, lipids. These are known as the eye cells. Eye cells. The sensors detecting fats are called eye cells, and they secrete a hormone called CCK, cholecystokinin. Lots of terms today. CCK cholecystokinin. CCK. Okay, so CCK, going back, CCK decreases gastric emptying. Okay, we'll talk more about what it also does in just a second. These acid sensing cells are called S cells. It makes sense because they secrete a hormone called secretin. Also decreases gastric emptying. And then these osmoreceptors are also feeding back. Not sure what that is, what hormone, but it also decreases gastric emptying. Now, there's a few other hormones that I want to talk about as well. There's chemoreceptors that also are working on these local pathways to, again, increase the pyloric junction contractions to decrease gastric emptying. Now, before I move on, I got to give you some anatomy real quick here. As always, I kind of tell you what you need to label for the final exam. This is one of those. You do need to know the anatomy of the gallbladder and the pancreas and how it relates to the duodenum. This is the first part of the small intestines. You can actually see here both deliver their molecules They're signaling molecules through a common sphincter of Oddi. Sounds weird, sphincter of Oddi, but it is named after an Italian scientist, Dr. Oddi. Okay, so you can actually see, again, the gallbladder sits right underneath the liver, and it contains bile. And then the pancreas is this glandular-looking organ. Sometimes it is yellow. I've seen it in certain species. It's kind of in this J loop within the small intestines. And it delivers bicarbonate coming from the stomach. Remember, we delivered all of that bicarbonate into the blood supply. It travels to the pancreas where it's delivered into the small intestines to neutralize acid coming from the stomach, and other pancreatic enzymes like lipase, proteases, and amylase. So you have a pancreatic amylase as well. That all gets delivered through the common sphincter of Oddi. All right, so let's zoom in to the pancreas. You can actually see within the pancreatic duct here, this is a typical, again, acinar gland. It has a bulb portion called the acinus. This is an exocrine gland, and duct cells. These duct cells, I think I'm going to put this all together for you. The duct cells actually are expressing CFTR. So individuals that have cystic fibrosis are not able to secrete that bicarbonate. And as a result, a lot of times you get these mucus plugs. And that really impedes any kind of digestive enzymes that are delivered into the duodenum. Even the mildest form of cystic fibrosis, they all have some kind of digestive enzyme dysfunction. A lot of times they have to take oral medication. So here's a much bigger picture. Let's talk about each of these, and that'll end today's. We still have about 3 more minutes. You do know some of this already. Let me put it together for you. Here's the stomach here. As a little bit of food enters into the duodenum, the eye cells detect fat, and they secrete CCK. CCK travels to both the gallbladder and the pancreas, and it triggers bile to be released, which emulsifies the fat. That helps to keep it into smaller fat droplets, so that lipase coming from the pancreas is delivered as well to break down that fat. It's a very elegant system, right? Eye cells are detecting fat, sends a signal to the gallbladder to release bile to emulsify the fat, and sends a signal to the pancreas to deliver lipase, an enzyme that degrades, breaks down fat. All right, acid. Acid coming from the stomach is detected by those S cells. It secretes secretin. It travels to the pancreas to release bicarbonate. It triggers the pancreas to release bicarbonate to neutralize that acid. Again, a very elegant system. GIP secreting cells are they used to be gastrointestinal peptide. They detect carbohydrates, and they send a signal to the pancreas to release insulin. Now they had to make it difficult. GIP used to be, again, gastrointestinal peptide. Now it's gastrointestinal, let's see, oh, now I'm losing it. It's insulinotropic hormone. Gastrointestinal insulinotropic hormone. So it detects carbohydrates, and then it actually releases insulin into the blood supply to trigger the pancreas to release insulin so that blood glucose levels can be lowered and taken up by those cells. Okay, I'll make sure that's correct. insulinotropic peptide. All right, and then again, G cells are in the stomach. They secrete gastrin, and that actually ramps up the system. So now you actually have a full picture of all the hormones. We will review this on Friday. We will have some top hat questions. We haven't had those for a little bit. So bring your top hat devices, and I'll make sure that I clear that up. Gastrointestinal insulinotropic hormones. All right, have a good Thursday. I will see you all on Friday.
Sorry, I had one more question about the presentation, about the
Sure,
video presentation. So when are we presenting?
You're not presenting. You're not,
Okay.
yeah,
Sounds
no
good.
problem. We don't have enough time for it. I've never had enough time for all the presentations.
Okay.
Yeah, it would be fun, but yeah.
Yeah, okay.
But yeah, I think we're good, and you'll just turn that in, and it should be fine.
Okay,
Yeah.
awesome. And I
Awesome.
sent it.
Oh,
our link
perfect.
to you, so,
Okay.
yeah, thank you for taking the time.
Yeah, absolutely.